Hello everyone, and welcome to a new episode and a new series. This new series will tackle the physiology of animals, organisms within the kingdom Animalia. Today is episode 67, where I'll be exploring animal development, starting with the fertilization of a new individual animal and going through its developmental processes until it reaches sexual maturity and can reproduce on its own to perpetuate its lineage and push its genes through time. The kingdom Animalia is awe-inspiring in its extreme diversity and uniqueness, even though basically all animals are built around the same basic premise. Now, I'll get into the physiology more later on in the episode, so for now I'm just going to describe animals in the most general terms possible. Fundamentally, animals are mobile feeding platforms. They're tubes that move around and acquire food. The acquired food is put into the mouth, or a hole at the front end of the tube, and it passes through the tube, which is the digestive tract, where the food has all of the nutrients extracted from it and absorbed into the animal's body. At the end of the digestive process, this tapped food is excreted as waste out of a second orifice at the end of the tube called an anus. These animals, with their basic tube body shape, have evolved all manner of limbs and protrusions and sensory organs to detect the world around them and to move within it. Their adaptations help them capture food so as to put it in the tube, and this sustains their bodies, at least until they can mature and reproduce and create offspring of their own. This is where the story of animal development begins. Because virtually all animals are sexual organisms, it takes the mating of a male and a female parent to produce a fertilized egg, or a zygote. When two sexually mature individuals meet out in the wild, they might engage in some kind of mating rituals or show-off behavior before actually performing sex. The male deposits his gametes in the form of sperm into the female, where the individual sperm attempt to navigate the female's reproductive architecture. They follow chemical cues to find and fertilize an egg. When sperm meets egg, the sperm first has to burrow through an outer jelly layer before reaching the egg's membrane itself. At that point, a chemical process occurs where the sperm binds to the egg's outer membrane, and this also changes so as to prevent any further sperm from binding. The single bound sperm is absorbed into the egg's membrane, and it deposits the haploid genetic material into the egg's cytoplasm. The sperm's haploid genome then finds and fuses with the egg's haploid genome, and this creates a fertilized diploid egg. An important thing to note here is that both the sperm and egg of any given species are coated with proteins that act as species-specific markers. The sperm cells express proteins on the outer surface of their membrane, and these act as ligands. They bind to stuff, and the egg cells express a complementary suite of protein receptors, things for these ligands to bind to. There are other kinds of expressed molecules that act as species-specific identifiers, like various glycoproteins, but these aren't of particular importance for this conversation. What's important is that these species-specific binders and receptors expressed on the sperm and eggs prevent the crossbreeding from organisms of different species. This is known as a prezygotic block to reproduction. It prevents the birth of hybrids, and thus it discourages species from blending together, and it encourages them to speciate and diverge. I talked about this phenomenon a lot more in my Intro to Evolution series, if you want to go check that out. Anyways, when fertilization occurs, the process of development begins. The fertilized egg will divide once, then twice, then a third time, and again, producing a dense little ball of cells. This process has some of the fastest rates of cell replication in the organism's entire life. The cells replicate on a time span of minutes, and this can produce many tens of thousands of cells in a single day's time. All of the increasingly small cells produced by this rapid division are called blastomeres, and they're part of a structure called a blastula. The blastula structure is just this thin little ball of cells. It's interesting to consider that these initial cells already demonstrate the beginning stages of differentiation in the tissues and the morphology that will result in the mature organism. So think back to the egg cell. It's a big, spherical shape, with cytoplasm filled with enzymes and sugars and nucleic acids and all this good stuff. Many of these molecules in the egg cytoplasm serve a regulatory purpose, and their position at some point in the egg cell cytoplasm is no accident. When the egg divides, the cleavages that form and separate new daughter cells from each other are accurately aligned, so as to properly arrange these clouds of regulatory molecules in the cytoplasm into specific organized groups of cells in the blastula. For example, a type of theoretical regulatory molecule could exist in a loose cloud in the upper left quadrant of the egg cell. Now, when the egg starts to divide, those daughter cells that are produced in the upper left quadrant will all have some of the original cytoplasm from that area of the original egg, along with all of those theoretical regulatory molecules. Now, when development continues and genes start getting expressed, these regulatory molecules will alter their expression in some way, and tissue differentiation will begin in earnest. These regulatory molecules control the transcription cascades and the protein expression of their cells, and this alters how they grow, what they do, what chemicals they express. It really does lay down the groundwork for tissue differentiation, which, as you'll see, is extremely important to the animal organism, to its healthy bodily function. So after a few days of rapid cellular division, the blastula has now become a mass of cells, all primed with a particular set of genetic regulators. This mass of cells typically has a cavity or a fluid-filled space inside of it that's called a blastocele. At this point, the blastula enters a phase called gastrulation, where the cells reorganize themselves in a careful dance, and they settle down in 3 distinct layers, called germ layers. This cellular reorganization establishes the body axes of the animal, where its head will grow, where its limbs will grow, how it will be oriented in space, that kind of thing. These newly created tissue layers, or germ layers, are the ectoderm, the mesoderm, and the endoderm. The prefixes kind of give it away, but the ectoderm is the outer layer of cells which wraps almost entirely around the gastrulating blastula. The mesoderm forms a secondary layer immediately beneath the ectoderm, and the endoderm tissue fills in the inner space, that inner cavity. The growing endoderm fills in the blastocele, but it forms a new internal cavity somewhere else, and this will become the animal's gut, or its stomach and intestines. This cavity will become the inside of the animal's food tube. The endoderm tissue will go on during development to form the inner lining of the body's tubes and organs, like the urinary tract, the digestive tract, and the respiratory tract. The endoderm also produces differentiated cells that grow into many important internal organs, like the liver, the lungs, and the thyroid, among several others. Now, the mesoderm, which is the middle tissue, meso, middle, the mesoderm grows into all of the internal stuff that's between our digestive tract, between the internal lining, and our skin, or the outer lining. The third layer, which is the ectoderm, produces cells that are widely differentiated. Now, as you would expect, the epidermis of the skin, our outermost layer, is produced from the ectoderm, as is the tissue that's lining the inside of the mouth and the rectal cavity. But the cornea and the lens of the eye are also produced from ectoderm tissue. Now, this might be surprising, but part of the ectoderm actually sinks deep within the developing organism and produces the brain and all of the nerves that innervate the body. Alright, so back to embryonic development. After gastrulation, these germ layers are in place, but the germ layers, like the mesoderm, they haven't created anything yet. None of the organs have actually been generated. This is about to occur, and it's through a process called organogenesis. The cells in the germ layers of the blastula divide, and they reorganize again, and they undergo more differentiation to become very specialized tissues. These specialized tissues form nodes, and they grow in size, and they become the various organs and structures of the body. All of these specialized tissues that form your organs all start as these little nodes or buds of specialized cells. Consider the chordate animals as an example. The chordate animals have a notochord, which is a rod-shaped mass of mesodermal tissue that forms along the back edge of the embryo. The notochord is like a scaffolding that helps organize other embryonic structures and coordinate their development. Some invertebrate animals retain their notochords throughout their life, but vertebrate animals, which are animals with a bony spinal column, their notochord eventually dissolves in the embryo. 3 structures are formed that run parallel alongside the notochord. Some of the mesoderm tissue condenses to form 2 long columns of cells called somites that run along either side of the notochord, along the animal's back. Now, as these somites form, they create kind of bulges. There are these rods running underneath the ectoderm. And so the ectoderm is kind of like a blanket that's being laid over like 2 logs of wood, I guess, if that helps you visualize it. And so you can see these 2 parallel ridges start to form in the ectoderm. Well, these ridges keep growing up on their own, and they curl inwards to meet each other and form a tube. This ectoderm structure is the neural tube, and it will go on to become the animal's spine and its brain. Like the vertebrate notochord, the somites are also temporary. The somites break up into differentiated chunks of tissue that become various muscles and various bones, and the outer lining of the organs, and the dermis of the skin. Early in this stage of development, cells can be moved around to different areas, and they'll adapt and blend into the specific differentiation of the cells around them. This is to say that transplanted cells will acquire the specialization of the cells around them, of the nearby cells. However, this plasticity will fade as development continues, and differentiating tissues begin to settle into place, and they become determined. A differentiating cell reaching determination can no longer change its differentiation. This is to say that when a cell becomes determined, or it reaches determination, it can no longer change the patterns of gene expression that give it its unique functional qualities. Remember that this differentiation began all the way back in the very beginning, with various regulatory molecules floating around in particular regions of the cytoplasm of the egg. At this point in development, the cellular precursors for all of the body's tissues have been positioned correctly, and their genetic expression has been specialized and sufficiently differentiated so that they've started making organs and bones and the brain and nerves and everything else. Everything is in place, all the tissues are growing, and they're swelling in size, and perpetually increasing in complexity as the cells inside of them divide and further differentiate into even more specialized kinds of tissues. In many species of animal, these embryos are grown inside of eggs, which are deposited by the female into a nest or into some kind of protective chamber, or a safe area where the eggs can grow and eventually hatch. The animals that do this, that lay eggs, are oviparous. They have an internal fertilization, which produces embryos in the eggs, and then the eggs are expelled from the mother's body. For example, birds are oviparous, and a big part of their life involves watching over and protecting their eggs in the nest. Many reptiles are also oviparous, but they tend to be a bit more detached. In many species of reptile, they'll bury their eggs in sand or dirt, and then they'll just leave, letting the young hatchlings fend for themselves. The embryos in these eggs survive and grow by feeding off of the nutrients that are contained within the egg yolk. The egg yolk is just a very nutritious layer of tissue that's grown in the egg to help the fetus survive, and that sustains the fetus until it can reach full development and hatch out of the egg. Other animal species are Ovoviviparous, meaning that the organism produces eggs like an oviparous animal, but instead of depositing the eggs outside of the body, the parent instead will retain the eggs in or on their body. However, even though they're still within the body, The eggs will still possess an egg yolk, and the fetuses within the eggs will feed exclusively, they won't take any nutrients from the mother. Other animal species are Oviparous, which means the eggs are produced unfertilized, and they get released into the surrounding environment where a male then fertilizes them externally. This is typically the case for aquatic animals because this is much easier to do in a marine environment, in a watery matrix. So there's a lot of bony fish, cnidarians, and echinoderms like starfish that do this. They have this kind of oviviparous reproductive tactic. Now some organisms don't grow in egg structures at all. The young are instead grown within the female's oviduct. These animals are called Viviparous, and there's two types of viviparous animals. Hemotrophic viviparous animals produce their young in the uterus, and they feed them with nutrients that come directly out of the mother's blood. What the mother eats and digests, the fetal offspring will also ingest. This nutrient exchange occurs through an organ like a placenta. Humans, and most other mammals for that matter, are all hemotrophic viviparous animals. This is how we do it. We have a placenta, and what the mother eats, those nutrients and those chemicals, maybe if she smokes or if she drinks alcohol, those chemicals in her blood can make it through the placenta and affect her offspring, her baby. Now, the other kind of viviparous animals are the histotrophic viviparous kind. These animals are grown in the uterus, but they don't feed off of the mother's blood nutrients. Instead, they literally cannibalize their embryonic siblings in the womb, eating up all of the nearby zygotes and embryos and anything else that is basically a little floating mass of tasty nutrients. This particularly brutal form of fetal development exists in a small handful of species, mostly sharks. In viviparous animals, like humans and most mammals, when the young are being grown inside of the uterus, they're going through a period called gestation. I'll talk more about gestation and the nitty-gritty biological details of gestation in humans in a future episode that I have planned. But for now, just understand that gestation is the period in development where the new organism develops from an embryo and forms a viable fetus within the mother. Gestation ends when the female gives birth. The offspring is born into the world, where it opens its eyes for the first time and takes its first breath of air in the big, wide world around it outside the womb. Okay, so I kind of want to change gears here for a minute. I've just gone through a brief overview of animal development as a process, but now I'm going to explore what's being developed and why. This means the animal's physiology, its limbs, its mouth, its eyes and ears, its body plan, and the associated behaviors and life strategies that the animal uses because of all of these morphological variables, all of these traits. To begin, I'll have to go back in time, to the very beginning of the animal lineage, before all of this variation and diversity in the animal kingdom had been evolved. The first evolutionary milestone that began to shape the emergent animal lineage was the emergence of multicellularity, more than 600 million years ago. The first multicellular organisms were masses of tissue that absorbed food directly out of the ambient water, and these eventually became the first sponges. The sponges, or the porifera, are the most primitive, simple animals. Some species have an extremely thin epithelial layer, which lines the inner and outer surfaces of their bodies, and their bodies themselves are mesohyl structures designed to be a mass of pores and channels. Water will flow through these pores and through these channels, and food particles in the water will get extracted and absorbed and ingested. Now, unlike most other animals, sponges only have a single germ layer, not three. And while there is some cellular differentiation, sponges don't really form true specialized tissues or organs of any kind. They're very, very basal animals. Despite their relative primitiveness, the sponges demonstrate a few things about the animal lineage, showing off the qualities that were there at the very beginning. These qualities include a primitive level of cellular specialization, programmed apoptosis as part of development, the use of flexible sheets of connected cells, and a capacity to recognize cells composing the self as opposed to cells from the non-self, or cells from other organisms in the environment, like maybe pathogens. From within the lineage of sponges emerged the first diploblasts. These diploblasts were the first animals with 2 germ layers in their embryonic stage instead of just 1, and these germ layers possessed cells that underwent significant differentiation to form true tissues. These diploblasts have an endoderm and an ectoderm, and they form the inner and outer lining, respectively, of the organism's body. Now, diploblasts are still simple animals. Think something like a ctenophore comb jelly, or cnidarian animals like sea anemones and jellyfish. All of these lack centralized nervous systems, and they also lack sophisticated bones and muscles. Instead, they have decentralized networks of nervous cells. Some species have inflated cells or structures that fill with water and act like a hydrostatic skeleton. And very thin muscle cell analogs help the organisms twitch and spin and numbly move their limbs around. They don't really have a whole lot of internal organs either. The outer and the inner epithelial layers are filled with a simple mass of jelly-like goo called mesoglia, and this gives the organisms a little bit of volume and some fragile structural integrity. From within the lineages of these diploblasts emerged an ancestral organism with a 3rd germ layer, which was the mesoderm. This new lineage with the 3 germ layers were called triploblasts, and this opened the doorway for the evolution of much more complex organisms. The mesoderm led to the evolution of complex and powerful muscle tissue, as well as very sophisticated vascular architecture and very powerful, efficient organs. Also worth noting is that the shape and the symmetry of the animals changed. Jellyfish and sea anemones are like domes and cylinders. They're shapes that are circular. These organisms are radially symmetric. They look the same no matter which side you're looking at. They have a top and a bottom, but they don't have a front or a back. They don't have a left or a right. Now, in contrast, triploblast organisms evolved a bilateral symmetry. Instead of being radially symmetric, these have longer, narrower bodies, with a definite right side and left side, and a specialized front side and a back side. This shift in body plan that gave the animals' bodies kind of a directionality allowed animals to move far faster than they could previously, and this sped up the development of complex food webs and ecologies. Also, the decentralized nervous systems of the diploblasts coalesced into a centralized nervous system in the triploblasts. The diploblasts can use a decentralized neural net pretty effectively. Consider their radial symmetry. Food can approach them from any angle, so having a diffuse neural net allows for maximal, localized responses in the food-capturing limbs, like a sea anemone's tentacles. However, in the triploblasts, with their bilateral symmetry and their mouth orifice at the front end, they weren't specialized so much for this kind of random, indiscriminate, just floating around and bumping into food strategy. Instead, their kind of organized, focused, directional body plan and centralized nervous system gave them a sensitive head region for optimally detecting and finding food. The body trails behind this head region, with the centralized nervous system rooting through the body, innervating the flesh and the limbs, providing sensation and mobility to the organism. Inherent in this equation is the cephalization of the animal, or the development of a head region in the first place. This head region, just literally a head, is at the front of the organism. It's the first part that goes wherever the animal is going. It's the first part of the animal's body that will run into food, and because it's at the front of the animal's body, it can see out in front of the organism unobstructed. It's for these reasons that an animal's head became a very important structure that's packed with all manner of sensory equipment and orifices for eating and respiration. The body is positioned behind the head, and the limbs on the body allow the organism to propel itself through the environment. It can search for food, chase after food, and run from predators. Consider the fact that these early triploblastic animals were all aquatic and soft-bodied. Within them, the Mesoderm evolved an internal, fluid-filled cavity called a coelom. This coelom created a hydrostatic skeleton that gave the animals stiffness and rigidity, and this in turn sharply improved their motility in the water. The coelom also provides an internal space for absorbed nutrients to float around and move throughout the body, as well as a space for the gut to move around and flex within the body's cavity, as the animal's outer meso and ectoderm layers will bend and twist. Now, if the gut was attached to the ectoderm through a solid mass of mesoderm, that solid mass of mesoderm would limit the flexibility and the mobility of the animal. So the coelom, which was this loose, fluid-filled cavity that kind of detached the gut, the coelom loosened them up, and it provided them the flexibility for this fast, twisting, wriggling motions. And you can really see this kind of twisting, flexible body structure when you look at coelomate worms. Okay, so where are we now? I've talked about a lineage of animals that has evolved a centralized nervous system. It's underwent cephalization to evolve a head structure, and they've evolved a coelom. These are the traits of the bilaterian animals, so named because they all display the bilaterian symmetry that I've described. And this is perhaps the foundational quality from which all of the other qualities are derived. Within this lineage of bilateria, two major groups emerged, the Protostomes and the Deuterostomes. In the near future, I'll be releasing episodes that will explore both of these groups in vivid detail. So for now, I'll just stick to the basics. The titular difference between the Protostomes and Deuterostomes takes place during gastrulation. In Protostomes, the hole in the germ layers that becomes the mouth will form first, and then the anus will form second. Furthermore, the coelom cavity in the Protostomes is formed when chunks of mesoderm cells grow into a large mass, and then the mass hollows out on the inside, either through apoptosis or through cellular migration. However the cells do it, they'll leave behind a void, and this void will get filled with fluid to become the coelom. In contrast, in Deuterostome blastula development, the hole in the germ layers that becomes the anus is the first to form, and the mouth orifice forms second. The mesoderm also doesn't form blocks of biomass that hollow out to form the coelom. Instead, in Deuterostomes, the mesoderm will bubble out from the gut and pinch off to seal in a cavity, which fills with fluid to become the coelom. So these two patterns of development form the same end structure. They both form a body with a coelom. However, they represent some of the earliest and some of the most basal differences between some of the largest divisions in the animal kingdom. This is the biology of animals 3, 4, 500 million years ago. When you start looking at the animal kingdom at the level of protostome and deuterostome, you start to see a wildly diverse and dynamic range of organisms, evolving new traits and losing old ones. For example, platyhelminths, or the flatworms, evolved to lose their coelom. The echinoderms, like starfish and the related species, they lost their bilateral symmetry, and they re-evolved a radial symmetry. There are other trends that were particularly advantageous, and this led to them being evolved independently across multiple lineages. One of the more obvious and basal traits of this nature is segmentation, which was evolved independently in annelids, arthropods, and chordates, and in some lineages of mollusks. Segmentation is pretty cool, and it's really trippy to think about, because it brings me back to the food tube metaphor. Animals are bodies built around a tube, which is meant for ingesting and digesting food. And as I've said, the food tubes have evolved all manner of appendages to help them move and sense their environment and capture prey or escape predators. Segmentation introduced a genetically efficient form of body planning, and as it was evolved, it was preserved. A small set of genes could be expressed at different points along the tube that composes the animal's body, and it could create repeating body structures, like legs or something. At each point of limb or appendage formation, The gene expression could be similar to that at nearby points, or it could be different. This gene expression, and the differences from point to point, from segment to segment along the tube, produces limbs with certain qualities, or appendages with certain traits. If you have a small toolbox of regulatory genes, these can then access the same general set of genes, and they can express certain genes a certain amount. And the end result is a controlled development of different limbs, like your arms and your legs. These limbs are generated with a similar set of genes, but the differences between them arise because of different gene expression, different proteins being used at different times and in different amounts. This leads to your legs being larger and stronger than your arms, and it leads to your arms being more flexible and more tactile than your legs. And it also controls the proper development of your hands and your feet at the end of the appropriate limbs. In contrast, consider an insect like an ant, which has 6 very similar legs. The gene expression to produce each leg is likely very similar to that used to produce another leg. But this gene expression in the ant's legs is entirely different than the gene expression in the mandibles and the antennae that are produced on the head, even though this involves many of the same genes. It just involves a radically different expression of those genes. The head, the thorax, and the abdomen of an insect are all body segments where specialization of gene expression has been streamlined. This is very much the same way that your head, your torso, and your pelvis are distinct body segments, with assorted limbs and appendages protruding from each one. For example, your ears and your nose extend from your head, so does your tongue. It extends out of the base of your mouth. Your arms extend out of your torso, and your legs extend out of your pelvis. The number, the size, the shape, and the function of all of these appendages are all based on this segmentation, on these slight alterations to gene expression along the length of the animal's body, along the length of the food tube. This, in itself, is a result of natural selection searching for an optimal biochemical efficiency. This method of body generation offers extremely high morphological plasticity relative to the number of genes that are involved. It also suggests that morphological changes over evolutionary lengths of time may not necessarily be due to mutations in the genome itself, but instead may be due to changes in the patterns of gene expression, like changes in the epigenetics. All of these limbs and appendages coming off of the organism's main body, or grown within its body, they all serve some purpose that helps the animal survive and propagate its genes through time. Sensory organs detect the ambient environment, and they help an animal stay aware of its surroundings. Light-sensitive cell structures can form eyes, and these help the animal detect light wavelengths and see the environment. Sound-sensitive cell structures can form ears, and these help the animals detect pressure waves in the air or the water that are perceived as consistent and recognizable sounds. They can be triangulated so that the animal can find the source of a sound. Then there are chemically sensitive cellular structures that can form tongues and noses and all manner of strange equivalent organs. Some animals have evolved really crazy sensory structures that give them huge fitness advantages. For example, some animals have magnetic sensitive organs that can detect magnetic fields, and this is useful for navigation during long migrations. Or if you're like a turtle and you're just wandering around the ocean and you need to know where you're going. Some animals, mostly predators, have organs that are sensitive to electricity, and they're capable of detecting the bioelectrical activity in the muscles of nearby animals. So try being a little fish, and you're trying to hide from something that can smell the electric activity in the cells of your muscles. I mean, good luck hiding from whatever predator is after you, you know? These are just some of the most common sensory organs in animals. There are many more, and they have a wide range of sensory abilities. So the way I've been describing animals, when boiled down to their most fundamental qualities, is that they're basically mobile food tubes. There's 2 things implied in this description here. One is that the animal can move, it's mobile. And 2 is that the animal eats stuff, it puts food in its tube. So how does the animal move? how does it eat stuff? Like everything else in the animal kingdom, the physiology is diverse. Limbs meant for movement are believed to be homologous in the animal kingdom, which is to say that the common ancestor of all limbed animals had a rudimentary set of limbs, and alterations to those ancestral genes and gene expression has provided all of the variety in limbs in the animals that we see today. There are lobed limbs, which are like little stumps that are just used to grip and crawl. There's tube limbs, which are like numerous long, thin lobes that use hydraulic pressure to pull the animal across the surface. There's slippery, twisting tentacle limbs, like those found in squid and octopus. And there are jointed limbs, like you would see in arthropods and chordates. Some of these jointed limb animals have even evolved wings, and this allows them to fly through the air. As for feeding, the appendages and morphologies and even the general feeding strategies are all just as diverse. Corals and sponges are filter feeders, which means they extract food by filtering it out of a liquid matrix. Baleen whales and flamingos are also filter feeders. They extract microorganisms out of vast gulps of water or bites of mud, respectively. Animals like sea cucumbers are deposit feeders. This means that they literally creep and crawl along the forest floor or the ocean floor or whatever floor of their habitat, and they feed from the layers of deposited detritus and waste. Now, in almost complete contrast to this, there are fluid-feeding organisms, like moths and hummingbirds, and these have tubular mouthparts that they use to suck up nectar from plants. Now, most animals, at least I'd imagine that it's most animals, are mass feeders. Humans, for example, are mass feeders, as are lions and cows and most insects. Mass feeders have evolved sharp mouthparts, like fangs or edged mandibles, and these can tear chunks of biomass off of their prey and bring them into the mouth, where they then enter the digestive tract. Lastly, and perhaps most importantly, animals produce appendages and internal body structures that are used for reproduction, for the perpetuation of their species. Now, simpler animals like corals and sponges are asexual, and they can reproduce by budding. However, they're a statistical anomaly because most animals in the animal kingdom are sexually reproducing creatures. They need a mate with complementary genitals and gametes in order to reproduce. The male animal typically has external genitalia, which it uses to penetrate the female and deposit sperm into her internal reproductive structures. And inside the female, the sperm fertilizes her eggs, and this leads to the production and development of a new individual animal. And with that said, I've come right around to the very beginning. That's about it for this episode, everybody. If you thought that I talked about animal limbs and mouthparts only very briefly, then don't worry, because as this series goes on, I'm going to talk about animal nutrition and their mouthparts. I'm going to talk about animal motility and their limbs and their muscles and how they move. I'm going to talk about animal reproduction in much greater detail. So we're going to talk about animal genitalia and mating strategies, and it's going to be fun. This is going to be a really cool series on the Kingdom Animalia. If you enjoyed this episode, then stay tuned for next week, where I'll explore the evolutionary relationship that animals have had with water. I'll dive headfirst into the biology of animal hydration. And as always, thanks for listening.
Would you like to support the BioLogic podcast? It's super easy. When you open a new episode, press the like button or share it with your friends. If you aren't subscribed, you should hit the subscribe button so you can enjoy a new episode every week. You can also peruse our official store, which has a ton of cool stuff like hand-designed t-shirts, hoodies, and stickers. All the links you need are in the description section below.